All right, folks, welcome back to the Serious Angler Podcast and back with one of our favorite segments here, the Real Biology, and one of our favorite people, Mr. Stephen Bardin, Fisheries Biologist. And I feel like I say it every episode, Steve, but back with another freaking heater, man. We got a pretty good one lined up.
Such a good episode. Um, we've got one of my good friends, one of my vendors. We've been talking to a lot of state biologists and really learning the process. But there's a whole other side of fisheries management that happens on the private sector side, and I brought one of those guys this time. And Josh Sakmar, man, what you're going to learn is there's some people that know too much. There's some people that just have too many things in their head. And whenever it comes to bass genetics and spawning, Josh is one of those guys. He just knows too much, but he's the manager of Red Hills Fish Hatchery. And Red Hills is where I buy largemouth Florida bass for my clients. So I hope you know that means they're good.
Yeah, he's a what we call the certified bass plug.
There we go. There we go.
He's the dealer. We'll preface this episode for people that are simpletons like me and still are a child in the mind, child at heart. Is if you want to hear what the process of building a bass version of Jurassic Park is like, that's what this episode's going to be about. So dude, I was giggling like a little kid throughout this one. It's pretty awesome.
But we were kids when Jurassic Park came out. I was a kid when the first one. You were a kid when the last one. But we understood it. We understood what they were doing, and Josh explains it well.
I still watch this. Like literally 2 days ago, I watched the first Jurassic Park.
I don't doubt it. I don't doubt it.
How much of a child I still am.
I don't doubt it. But Josh explains it well. I think that it was clear what he was discussing. And I think also, I mean, if you're a fan of, let's say, F1 largemouth, I think there's some information in this that would really help you make better decisions on when you should or should not use something like that. If you're a fan of Florida largemouth or northern largemouth, there's just a lot of nuggets in this one. I'm excited for it. I've got a lot going on in what we're doing personally right now. I'm headed to Chickamauga this week to do a habitat project with Major League Fishing. Have you been out on the water much, Bailey?
I've been out a few times we got home. The problem for me, obviously still being from the kayak, is we just have been, like the most of the country, and especially us here in Great Lake country, just hit hard with wind. And so with that and our water temp still being high 30s, super low 40s is, the fishing's not hot and heavy. It is on certain lakes, but usually those lakes are the ones that are heavily affected by wind. And I just don't feel like being in 3-footers on water that is high 30s. So like I'll be out there if it's not too bad, but it's just one of those things that's risk versus reward for me right now, where it's, if I had a boat for sure, I'd be out every freaking day. But
I see the tan though.
Say what?
I see the tan. You've been out on the water. I can tell.
Well, yeah, that's from Texas still. That's from Texas and California. I still got that sunburn.
Okay.
Yeah, I still got the raccoon eyes. I'll have it till November now. But no, I've been out for a little bit, playing with some smallmouth pets of mine, and I say pets, they're community whole fish. But either way, it's fun nonetheless. And it's that time of year here in the North, Steve, where we call it the KVD season because everybody can be a hero. That's what we love. We love dumb fish. That's why we live in the North, is because we like our dumb fish. And so it's a great time of year to be out here fishing in the North and catching many fish, and they're all fat and happy. So it's you're always having a great time regardless of where you're going fishing. So if you're in the North, go fishing. It's a great time. Get the boats out, And
if it's too cold, listen to this episode with Josh Sakmar.
And if it's not too cold and you're fishing, still listen to this episode.
Yeah, it's a good one. It's a good one.
I absolutely love the feedback that we get, like DMs from people that show like where they listen to. Like we have multiple people that will send us episodes, and whether it's them graphing on the water and they'll listen to the show, or literally they have their phone at their feet and they're listening to the podcast while they're fishing. I absolutely love that. That makes me very happy. Yeah, so super cool, man. But I don't want to waste too much time because this episode I feel like is pretty juicy. And you mentioned earlier you think he clears it up pretty well, and I agree. I think he does. But if for whatever reason in these episodes, if people have questions on it that wants more specifics, comment in the YouTubes, send us an email. send us a DM on social so that we can then clear it up for you on a future episode, or when we have that guest back on again, we can dig deeper into it. But either way, I mean, the point of me being here is because I'm the simpleton. I'm the guy that doesn't understand. So feel free. You're not alone in that. So if there's something we want to explain further, we can put on a lake hotline, things of that nature. So if you have questions, feel free, let them rip. But I think Steve, let's get Josh on here, man.
Yeah, let's do it.
All right. Without further ado, let's bring him on. So, all right, folks, we're here with our next guest here on the Real Biology, Mr. Josh Sakmar. Steve, we got another one lined up for us, and I don't think the folks are ready for what's about to happen on this episode.
Banger after banger. That's what we're going to be known for. We are just putting out the hits. This one is going to be, I think really fun. Josh is the director of biology and infrastructure at Red Hills Fishery. Before that, he was with the infamous Florida Fish and Wildlife Conservation Commission. And so he's got some state background. He's got some private sector background. He's the guy I buy bass from. So he's legit.
That's a line right there. The guy that I buy bass from. So you're saying that Josh is the bass plug?
Yeah, he's the man. If you have a private lake, public lake, I don't care what you got. You need bass, you call Josh. He knows everything there is to know about their genetics and growth rates, and we're going to get into it today. Josh, thank you so much for being on the show, man.
Well, thank you guys for having me. I realized when you said FWC that I've got a very old FWC shirt on because it's cold out, so I had to kind of cover up the logo there. I don't work for them currently.
Yeah, let's just disclaimer right in the beginning. Josh, in all of his comments, do not reflect FWC.
Absolutely not.
I feel like we need to just put that on each of these shows.
Exactly. Well, let's start with FWC. What position did you hold with them, and kind of what was that gig like while you were there?
So towards the end, I guess I was the assistant hatchery manager / manager in training for the Florida Bass Conservation Center, which is in Webster, Florida, which is smack dab in the middle of the state. And when I was there, it was one of the only parts of the state that did not have consistent cell phone coverage, way out in the middle of nothing. But when I left, that was my position, but I had worked up from a volunteer. So when I decided to go back to school to work on fisheries, after, endeavoring in some other areas, the first thing I did was volunteer with groups. And I went out with some saltwater guys. I went out with some freshwater guys. And somebody suggested that there was this brand new high-tech hatchery that I should go volunteer at because, hatchery operations are something that I think anybody that works with fisheries, as far as stocking and sport fish go, should have some familiarity with. But so I went out there. I was not expecting to work in a hatchery. I wanted to be on the water. And after a few weeks, I kind of realized that, working in a hatchery, especially a state hatchery that's kind of on the cutting edge, gives you a lot of opportunities to do cool work because everybody works with a hatchery at some point. When you look at state agencies, especially, if you're working out of a regional office, that means you're working in your region. And typically when you start off, that means you're doing a lot of the grunt work outside. If you're on the saltwater side, you're pulling nets in the blazing sun all day long. If you're on the freshwater side, you're typically on the front of a shock boat, getting attacked by mosquitoes and bugs and everything else in the blazing heat. But in a hatchery, you have the opportunity to work with fish all day long in a controlled environment. And like I said, everybody eventually wants to work with a hatchery at some point because they need fish. And on the other side, if you're at a really good one, a lot of universities and other groups want to work with a hatchery because they want to do research. So I started off volunteering, and I volunteered for, probably 9 months before I got what's considered like a part-time tech position. And then that became a full-time tech position. And then once I got a biology degree, which is what they required for fisheries, I got a biologist position, and then I got a master's while I was there and just kept moving my way up the food chain. So I did that. I was there for about, I think, 13 and a half years doing all that.
Josh, you did it the prototype way that we tell students to do it. Volunteer, work the
Volunteer.
system, get your degree, become a biologist, get your master's, advance through the system. So if you've listened to Real Biology, we've talked about this before. If you're a student, that is how guys like Josh and I made it through the system. There may be shortcuts now, but I doubt it because fisheries is still old school.
Very old school. And volunteering, I mean, I'd volunteered a ton of different places, not just with Florida, but prior to that, I was in Missouri, so I volunteered with the U.S. Fish and Wildlife Service. I volunteered with Missouri Department of uh, MDC, Missouri Department of Conservation. Sorry, every state's got a different name.
Yeah.
And just kind of went around and tried to figure out what I wanted to do. And if you put volunteer on a resume, if you've got a couple lines of that, especially in fisheries, that goes a long way when you're trying to get an entry level position, because that means A, you've got some experience, and B, you've got the desire and the passion to do the work. Because at the end of the day, especially when you're younger, you're not going to be making it very far unless you got passion, because the money's not going to carry you that far.
Yeah, love it. Perfect way to start this. So, Josh, while you were at Florida working in the hatchery system, what species did you guys focus on mainly?
So it was the Florida Bass Conservation Center. So at the time, it was largemouth bass, Florida strain. But then we also worked with, I think in production during any given year, we worked with 5 to 7 other species, and then did all sorts of random stuff like American shad. We held some redfish at one point in time for a brief period. But the cool thing about Florida is that there's so much diversity. Um, and being a freshwater and a saltwater state, and having hatchery systems that worked pretty well together, you could be working on largemouth bass one day and then be going over to the coast to help with some sort of snook operation the next day. So it was pretty cool. It was a great way to get some diversity and some training.
Yeah, and while you were there, you did make a couple publications. Like you said, you had research with universities, and you had a couple publications, and one of those was on some DNA technique, right? Molecular technique.
Yeah, so we contributed to, so at the time when some of this was going on, I wasn't getting authorship on anything necessarily,
Right.
but I was able to assist with a lot of projects. And as I moved up in my career, I started to be able to get listed as an author on some things. But we did do a lot of genetics work. We assisted with surveys of the state. The goal of the hatchery itself, as the Florida Bass Conservation Center, was to conserve and protect endemic populations of Florida bass. So a good chunk of what we did was based on genetics, because if you lose the Florida bass in Florida, there's nowhere else to go. That's it. And we all know from a fisheries standpoint, if you're looking at making large bass, you need to have some Florida genetics in there.
Yeah, we're going to get into that in big detail, but genetics is really becoming popular today, and you were doing it maybe before it was extremely cool. So let's tell the audience a little bit about how we collect DNA from a fish, and then what does the DNA even mean? Like, what does this mean?
Sure. Well, there's multiple ways that you can get DNA from a fish. The old school way was to do biopsies, where you would take, you'd try to get a sample of the liver, and then send that off for what was called, you'd run gels, so electrophoresis. And that was done in the 80s. Yeah, that's that's a very old school way of doing it, and not very accurate, and not too many people do it anymore. But it's basically, you take DNA and you put it into an Argos gel, and you send a current into it. And based on the movement of the DNA through the gel, then you can make some what's the word I'm looking for? You can make some assumptions about what is actually going on from a genetics perspective within the fish. The problem with that is that it's an old school way of doing it, and it's not the most accurate way of doing it compared to what we have these days. So when I came in, microsatellites were the big thing, And when you look at DNA is like the best analogy I have is it's like computer code, a little bit more complicated. But if you look at it as ones and zeros, and you look at what backs up your computer, it's huge strands of ones and zeros that allow us to do kind of what we're doing today, which is, having a podcast, from various different regions within the U.S. So there's huge strands of ones and zeros, and just like that, DNA has got huge strands of proteins, and there's four main types of proteins. So as opposed to ones and zeros, you have basically 0, 1, 2, and 3. When you look at microsatellites, you're looking at specific sections of those numbers, and then within individuals, that DNA is standard in certain areas and it's variable in others, which allows, like I'm 6'2", I've got blonde hair, but that's going away now. You guys have got differences in your appearances. Well, it's the same thing in DNA, where you would have some specific structure in certain areas, and other areas you have variation. You can identify those areas in variation and look at how those strands are placed and how they're designed. Then you can take a look at differences across populations. And that's what I came into, is microsatellites. But we've also moved into things like what are called single nucleotide polymorphisms, where as opposed to looking at chunks, I know it's a lot. I still trip up over saying that. But as opposed to looking at chunks of DNA, you're looking at individual zeros, ones, and twos and threes, and looking at variation there. It's a very the modern approach. It's very accurate, and it's what we call high throughput, which means you can do it fairly quickly in a week if you can get it set up right, as opposed to some of these other techniques which take a good bit of time.
Okay.
Hopefully, I did that well.
I think you did that really well.
I think there's one word in there that threw me off a little bit. I don't know. I can't even repeat it. I don't even know what it was.
Single nucleotide polymorphisms.
Yeah, that one.
You just say it in a song. You just say it over and over again.
Let's see, Bailey. Let's try to summarize this. DNA is a code, and all species have a specific code, but then there's sections in that code that give you different traits for that individual. So Josh started out by looking at sections of code to identify what types of bass were present, and somehow also could see the sections where traits were different and knew that was different from a species. And then now y'all are looking at smaller single pieces of that code to identify species. So you're getting more accurate, and it's faster.
Yep.
And all we need to know is, what is the acronym for the newest type?
SNPs, SNPs.
SNPs, Bailey, we got that. Me and you can handle SNP.
SNPs.
Yeah,
I feel like I'm back in 10th grade science, learning about chromosomes and everything right now.
Hey, no, this is what's perfect. Now, whenever we have other geneticists on, we say, hey, how's y'all's SNPs? And then we look like we're in the know.
That's right.
Okay. So you were doing microsatellite at the time. Maybe this is a leading question. How many bass species were in Florida based on microsatellites in the early 2000s when you were there, or mid 2000s when you were there?
Species?
Yeah, let's just do species. One. And what was that species called?
Largemouth bass.
Yeah. And then, I know you're not there now, but if you were there now, how many species is there now?
Based on the current nomenclature, there's two.
Correct.
There's the largemouth bass, and there's the Florida bass, which are two distinct species now. And
so the early work that you and y'all's team were doing, we know it wasn't just you by yourself, but the teams basically set the stage for this. You guys mapped the genetics throughout the entire state.
Yes, that was a PhD by the name of Brandon Barthel, who's still the geneticist, the black bass geneticist for the state of Florida. that's something he had done for his PhD work, was to actually go through and map differences in Florida bass populations across the state, which kind of set everything up for everybody else from a management perspective. On my end, my job was to uh, produce fish, um, ensure that they were Florida bass, but then also based on his work, we had different regions within the state, so different populations. So we had to hold different, what we call broodstock, or the parents, from different regions so that we could put those back into those regions from a hatchery perspective. So even within Florida bass, when they were just a strain, there's different populations of Florida bass that we wanted to protect and that they still work to protect within the state of Florida.
Very interesting. So you're doing the microsatellites, and so you're doing it regionally. Are you able to see trait differences throughout the state and looking at the microsatellite?
So though we didn't really have any quantitative data on it, like we never really ran a study, but after having, so we had 3 different regions within the state that we worked with, 2 of which we held at our facility in Central Florida. So we had what were called the St. John's fish and then the West Coast fish. And this was all based on watersheds and drainages. From what we could see, looking at not only the parents but the offspring, it looked like West Coast fish, which is the Gulf side, those fish often were squatter and taller. They were a fatter fish. And the St. John's fish, which is kind of the East Coast side, tended to be a lot longer and skinnier. So they had a bigger frame, but they didn't seem to pack on the weight for that frame like the West Coast fish did.
And the goal at that time would be to separate those so that you could then produce fish if you needed to stock into those environments, maybe after a hurricane, or I don't know why you would stock, but you would stock, but you would keep them separated. Was there ever discussion of maybe we like this west side fish morphology a little bit better, so should we introduce that onto the east, or was that always let's keep them separate?
It's always let's keep them separate. And the mission of the state of Florida, and I'm not going to speak for them, but the way that I saw it when I was there was to conserve. Again, because if you lose those populations in the state, then you lose them forever. So though the West Coast fish might have appeared to be a better fish as far as the angler goes, from a conservation perspective, it was a separate and distinct population, where if we introduced those to different populations across the state, there's no way of predicting what could happen. You could hurt the population to the point that you actually get worse fisheries. And since Florida is a hotbed of invasive species and moving things around and things that aren't supposed to be there, that was something that I don't think we really wanted to do at the time, was to start mixing up genetics for something that was really special to us.
Now, would y'all consider those at the time subspecies or just regional populations? Like, how would y'all define them?
So we called them those areas genetic management units, or GMUs. So at the time you had one species and then you had 2 different strains, northern bass and Florida bass. And then amongst the Florida bass, you had different populations that made up that particular strain.
And then what did y'all call the fish that were in like the northern panhandle area?
Panhandle fish. There was a lot more diversity up there. When going back to why we would stock fish, there was 2 reasons really. If you had a lake that was started over, which there were a few that we worked on, you would go back in and stock with forage and stock with bass. But one of the big things we worked on was to conserve the genetics that were endemic, which means that was a whole different reason to stock. That was a genetic reason. So we would go in and try to flood a population that had northern genes. We'd try to swap those out with native genes. because all the way down into South Florida, we were seeing genetics that shouldn't be there. And that's because, over the course of time, we had moved fish around. We had stocked fish from up north, and this was long before my time there. But when people didn't know that there was that big of a difference. So in order to protect those populations, we would go stock on top of existing populations and try to swamp out those northern genes.
Man, that's so interesting. So today, SNPs have become more popular in genetic analysis of bass, and they're more accurate. And so, do you see the Florida bass home range increasing or decreasing based on the more accurate genetic we're seeing now? Like you're saying, there's some northern fish were moved around, but what about like our initial home range analysis? Has it changed?
It has changed. So it's changed within the past 3 years. When everything was one species, we were working based on existing data. And within the past 3 years, new data has come out that's based on the most recent technology and based on a really broad survey. And that has actually done two things. One, it said we no longer have one species, we have two. And then the other thing that did was to extend the home range of Florida genetics outside the state of Florida, actually all the way up to South Carolina. So when you look at kind of that coastal strip that goes through Georgia, and it's really got to be coastal up into the Carolinas, those are now, according to the current standards, native Florida genetics, as we would call them.
Wow. I mean, come on, Bailey. We're changing science up to 3 years ago. This is pretty cool. And the SNPs and the microsatellites, are y'all still using liver samples to do those collections, or what? How do you collect that DNA?
No, that was for stuff that the early on stuff. What we do now is we can take a piece of tissue from a fin, and in some cases we can do a buccal swab, which is a swab in the mouth. The fin seems to be, at least with the technology we have right now, it seems to be better at giving us quality DNA. The swab works in a lot of cases also, but if you really want to kind of hedge your bet that you're going to get results, right now it's a little bit of tissue off the fin. And that's really only the size of your pinky nail. We really don't need that much in order to get quality material out toward an analysis.
So we went from a liver and not great analysis to microsatellite and fins, and now we're at SNPs. Is there another layer that's coming? Do you hear scuttle that there's another way to do this? It's going to be better?
There's probably, so there's different variations of all of this. I always tell people that fisheries biology from a genetic standpoint is probably 20 to 30 years behind human health. And that's mainly because of cost. So if you look at human health and look at what's going on these days, that's going to be applied to fisheries probably in the next 20 years when cost comes down. So there's all sorts of things that you can look at. And really, like we were talking about, I think a little bit before, this world has blown up, and especially in human health. I mean, there are all sorts of various projects. I was reading one about a company that brought back a dire wolf, which has been extinct for 10,000 years.
Seen that.
Yeah, so there's
they're
all
doing woolly mammoth too. Not to sidebar, but that's
yeah. I saw woolly mouse, woolly mammoths coming. So it's crazy to think of what you can do down the road from a conservation perspective. It's really, we're just on the cusp of doing some great things, I think, that are good for the species and good for humans when it comes to conservation.
Here's my question, and I don't mean to take this on a total sidebar, but when I seen this post and the accuracy of it, like it's actually a real thing with the direwolf and the mammoth, and I literally, not that I'm just a Jurassic Park fan in general, just a nut about it, but the line in the first movie played in my head of where I'm trying to remember the guy's name, but he's just like, He's like, did your scientists ever think about, what they could and then if they should? And then
Right.
that played in my head, and I'm like, where's the line here? Like, what are we gonna plan on not bringing back? Like, some
And
things are maybe left better in history, like a T-Rex. You know what I mean?
yeah, and that's all stuff that's going to have to be, worked out from a tech standpoint. Yeah, we can do all that stuff. From an ethics standpoint, do we want to do that stuff?
It makes me
Um,
want to just, the content creator in me makes me want to just go sit out in the tree stand and make a funny reel of just like in 2040, you're sitting in the tree stand, there's a T-Rex walking through the woods. Like,
yeah, a horde of velociraptors you're running from.
yeah, you're
Yeah,
competing with dinosaurs for deer. Like, yeah.
Well, Bailey, that's actually the perfect segue, I think, because Josh, you left Florida, and in Florida, your job was to conserve black bass species, and you came over to Red Hills, a startup at the time, and to
Mhm.
be the head of biology. And at Red Hills, you kind of are asking the questions of what can we do and what should we do when
Correct.
it comes to growing trophy bass. And so I know it's not Jurassic Park, but what is the difference in approach from your former position to your current position whenever you talk about hatchery management and what y'all are doing, selecting for and looking for?
Sure. So like I said before, within Florida, it was all about conservation and protecting something. Here, it's the same mission, but we have a little bit more flexibility in addressing some of those questions because, for one, we're not within the state of Florida, which means we're not as honed in on protecting a native species. But two, when you work for a state hatchery, we were, like I said before, you're working with all sorts of other stuff. I mean, we were making channel catfish, hybrid stripers, bluegill, all sorts of different species, which means your attention is getting dragged into all sorts of different projects. At our place right now, the only thing we focus on is black bass and specifically largemouth and Floridas. So we can take this whole property, this entire facility that we have, and run experiments, dig into questions, do general production. That's probably the biggest difference is that we can focus exclusively on what are now two species as opposed to being, our attention going elsewhere at certain seasons. On top of that, when you work for a large agency, not only is your attention getting distracted at certain times of the year with other important projects, but you're also always competing for funding. You're competing for space, and again here because all we do is largemouth in Florida is all the funding goes directly into that research, and it's all here to support the mission. And on top of that, we have the folks that own the company, the family that owns it, is very keen to do good conservation work on black bass, which makes it even more streamlined when it comes to getting things done.
No, I think whenever it comes to just producing quality fish, I mean, Red Hills, you guys have it down to a science. I've got so many questions about this. I guess we should continue on this genetics topic and go through how you looked at Florida and, well, more specifically, yeah, Florida bass. How you looked at Florida bass genetically in a different lens whenever you got to Red Hills. Like, what were the things you were looking at for those fish specifically in
Sure.
learning DNA?
So we were interested in things that the angler is interested in to start, which is growth rate, ultimate size, aggression, and gender. We all know that the females get bigger. So being able to rapidly identify female fish was a very important thing. And potentially, though we're not there yet for various reasons, we started to investigate the ability to produce all female fish. The other thing was ultimate size. How big are these fish going to get given the time and resources that folks can put into them? So, there's northerns, for instance, or sorry, largemouth. I still get confused. Largemouth, are going to top out probably at around 6 or 7 pounds for a good fish. Where we know the Floridas and the world record gets up to 22 pounds. And 15-pound Florida is not unheard of, and that's a very good fish. It's not super common, but that potential just isn't there with largemouth. So we started looking at those two things. And my thought was ultimate size is going to take forever to figure out because you need a bunch of samples. Gender is going to be very easy to figure out from a genetic standpoint because all we have to do is basically cut open a bunch of fish and say whether or not they're male or female. So we had a short-term project and then we had a long-term project. After a couple of years of working on both, which was a lot longer than I thought it was going to take for the gender, we realized that gender is not as straightforward as we had contemplated in the beginning from a genetic standpoint with bass. And we were able to predict, this is probably getting way into the weeds, but with it from generation to generation, it seems like the markers that had worked in generation 1 would not work in generation 2, which means that we could predict something, usually with a high degree of precision, in the first generation, it would all fall apart in the second one. So we did multiple studies like that because at first I'm like, well, obviously it's a data problem, right? Obviously, I don't have the ability to line up columns on a data sheet properly, so let's start over again. And we did that 3 times with the same results. Every time we came to validation, everything would fall apart. And concurrent to that, we're also working on the trophy side, trying to find markers for fish that get big. And all this is about markers. It all goes back to single differences in that code. So we were trying to get samples of fish over 8 pounds, which is a challenge. And when you're doing this kind of work, you need hundreds of those samples. We were running that at the same time. We got to the point where we had about 300 fish over 8 pounds that we decided we would run a pilot study on. And at the same time all this is going on, the state of Texas is also doing an independent study on their own, basically looking at the exact same stuff. Luckily, Texas is a great state, and we are friendly with the folks in Texas, so we were able to have conversations about what's going on with their project versus ours. When we ran the trophy stuff in what we considered a pilot study, we found 12 markers that not only predicted ultimate size within our data sets, but highly predicted ultimate size. So statistically, it was very, very easy to pick out. And on top of that, I believe 5 of those markers, Texas also got. So you have 2 different independent studies from 2 different groups that are overlapping. That was something that I thought was going to take 8-10 years to figure out. At the same time, Texas was also doing gender work and was having the same problem about their tests falling apart. So it completely flip-flopped on me to where we have now 12 markers that we consider highly predictable of ultimate size eventually, but the gender stuff we're still working on, trying to find markers. Now we do have some other techniques we use to rapidly identify females based on some physical factors. That's very good, but ultimately you want a genetics test because if you do a genetics test, you can do 20,000 fish in batches. The other way, you're doing one at a time over and over again.
So I don't want to oversimplify this, but for the audience, what I just heard Josh say is, in Florida, they were looking at this big parts of the code and they were trying to find what matches to say this is a Florida bass or a genetically specific, regionally specific Florida bass strain. And in his new role at Red Hills, he still has that to identify Florida versus Northern, but also he's now looking at those other sections that are the traits that change from individuals. And you found 12 of those sections that if a fish possesses those 12 in the right order, that is prediction that fish will grow over 8 pounds, especially if it's a female. What's the biggest male you've ever seen?
I've seen males that are in that 6+ pound range.
Okay.
But and confirmed them.
To get to an 8 and plus, I mean, definitely being a female helps, but it's almost always a female. So this code, these what do we call those 12 things?
And we call them 12 markers.
Markers. So these 12 markers, if a fish has those, it has a better odds of getting to over 8 pounds. And that's the sellable product. That's what you guys have today.
Yeah, so it's and we don't know what that code actually codes for from a genetic standpoint. It could code for being less aggressive. It could code for better health. That's all a step further than where we're at. All we have right now, which is pretty good, I think, is stating that 8 pound plus fish have some genetic commonalities that are not seen in general populations. And we've identified those genetic commonalities not only in a study, but we've identified them in our brood stock and have selected those brood stock to spawn together. So that over the course of time, not only are we ensuring that the offspring have those markers, but we can selectively, pair individual parents together to compound those markers over generations. And the thing is, this is why I think it's all cool, is that if you come into our facility, people are always kind of disappointed that we don't have like a bunch of 12-pound fish running around. Right. We don't need 12-pound fish. If we have the genetics, we know that those offspring are going to have the genetics and the likelihood of genetically reaching a certain size. Whereas, the old school way to do it was you take a 12-pound fish and then you breed it and you wait for the offspring to get to 12 pounds. And you do that maybe 3 or 4 times in your career and it's time to retire. Here we can do that basically every 16 months. So you can compound that very quickly over a much shorter period of time. And you can ensure that all the offspring have what you want at the end of the day.
So basically, what Red Hills is able to do is create what Toyota ShareLunker program is, which is selecting for growth, overall size, and instead of using broodstock captured from the wild that already exhibit those traits, you're able to use their offspring that carry those same traits at a smaller size, easier to handle, all the benefits of that, and produce high quality offspring, which is what Texas does in the Lone Star bass, where they've now replaced their Florida bass with ShareLunker offsprings that they're spawning together to create Lone Star bass. So you guys on the private side offer the similar technology.
It's a very similar approach. I think don't take this the wrong way, Stephen. I think we're probably a step ahead of Texas, simply because of our ability to look at all of the offspring that come off of a production line. And based on those genetics, we can determine which ones are going to go on to the next generation of parents. But yes, it's a very similar approach and idea to what Texas is doing.
Yep. And we're speaking, let me clarify, and maybe I'm wrong. We're speaking specifically about Florida bass.
Correct.
Okay.
Yep. Yeah, the trick with the northerns, the first thing is, what's a trophy northern? With Florida's, it's pretty straightforward. You just try to find the biggest fish you can get. But you can't do that with northerns because you don't know where the cutoff is. On top of that, it's much more difficult to collect northern tissue samples because they don't have the programs in a lot of areas in the northern states that they do in the south, i.e. ShareLunker or trophy catch in Florida. You don't have the same emphasis on big fish from a scientific standpoint. So though we would like to get into that, and that's something we're currently kind of behind the scenes trying to queue up, it's a much more daunting task. It's just not as easy to do.
Hey, well, maybe I know a guy.
Yeah, maybe. You do know a guy. Bailey's counterpart on the other serious angler, Andy, is a guide, and I hear that he's seen a couple largemouth that are quality size.
He's more of the smallmouth guy because he's on Lake Erie all the time. If you need some big largemouth, I know some people. I mean, there was a 12 caught in the Finger Lakes last year.
Really?
Yeah, state record was broken, shattered, I should say, actually.
Yeah.
By over 1.5 pounds. But no, we have actually a very healthy amount of 6+ pound largemouth in the Finger Lakes. Couple already been caught this year that are over 8. So like there's some pretty big ones. If you need any, I don't know what's involved in the testing, but if you need people to do it in the north and send you stuff, I know a community would be happy to do it.
I think that would be very interesting, not just from our perspective, but from a conservation perspective too. Because one thing that Steve, I don't know if you know of anybody that's really done a survey like it's been done in the southeast, but largemouth bass, the geographic region is much bigger and it encompasses many more watersheds than the Florida bass. And I don't know of anybody that's really gone out there and tried to assess that.
See, this may be my ignorance, but whenever I think about genetic work on northern bass, most of it falls back to Great Lake systems. Most of it. I mean, that's the stuff that I can really think of. Or let's think of the states of Iowa. The state of Iowa has a lot of good research on their water bodies, but I don't know that they've compared them in the same way that you guys were doing in Florida, where we're looking at regionally what are those differences. And I think part of that is because of the overall size. If we were seeing northern fish growing to 15, 16 pounds, I think you would, on the angler's side of it, you would have more anglers pushing for, let's understand why these specific water bodies. So you take a cayuga, that, correct me if I'm wrong, Bailey, y'all's smallmouth bass record was broken. Your largemouth bass
record, largemouth and smallmouth out of cayuga broken 4 times in the past 4 years.
So there's something special happening there.
Yeah.
There's nothing being studied about it, which I'm going to hold my tongue, but there's nothing being done.
I would attribute a lot of it to new forage with the introduction of gobies. I think whenever you have a new forage being introduced, that can really change a fishery. But if we could get samples, Josh, tell Bailey how many you need. Send him a kit. Say, here's how you do it. And I think we're 6 months away. We're 6 months away from running a panel
to do that.
Yeah, absolutely. We'll definitely talk about doing that, and that's what's always interested me is that like what you're saying right there. There has to be a genetic difference in the fish that you see that have been breaking records versus what's in Kansas or in New York. It's different watersheds that in some cases are separated by mountains. There's the natural transfer of genes was not going to happen. So there's got to be some difference there. And once you start to map those things out and kind of look at those differences, then not only can we hone in better on conservation, but we can hone in on what makes fisheries better for the angler at the end of the day.
We're all for that. I mean, if that's something you need, we got a whole herd up here. Perfect. That is willing to participate and be a part of that, because we've been wanting that for years, and we've noticed this trend coming, because not only obviously angler skill levels getting better and learning how to unlock the lakes better, but it was probably almost 5 years ago when I caught a fish on Cayuga that it was a largemouth that spat up a goby, that I knew, okay, this is about to boom. And I wasn't the only one there was many before me that realized that as well. And so it's now every lake that has a canal system that attaches to Lake Ontario has gobies in it, which is actually really cool to see, because the lakes are changing a little bit. The size is going way up. I mean, it's just little chicken nuggets on the bottom for them to eat. So it's, but it also is like, okay, how's it impacting the fisheries too? And I think anglers, our community up here, I could tell you across New York, is hungry for that kind of information, or at least to be done, so we can learn and understand and better take care of our fisheries. And yeah, that's a whole other conversation that Steve knows about that could be had another day.
Well, we're not talking about something that's extremely expensive. And, like Josh said, we had 300 fish over 8 pounds, whenever he did the initial largemouth or Florida bass study. So if it's not terribly expensive, and we could probably find some partner groups in the north and northeast to work on this. And maybe we'll chat offline because I think we can come up with something cool. But I mean, what does it cost, Josh, roughly to test one fish and figure out genetics?
So there's two different tiers of analysis. The first tier is kind of the exploratory tier. That gets a little pricey. That's like those 300 that we looked at. We had to look at um, you know millions of pieces of data in order to draw some inferences there. Once you have that analysis done, and say you refine it down to 12 markers, then it's fairly simple. You can go on our website and purchase a kit that'll give you, the trophy markers, the diversity, how fish are related within a population, the strain, and all that comes in a very, very detailed report. That's for 35 fish is $1,500, which for most folks is really probably what you spend in feed on a decent sized farm pond in a year. But looking at it from a baseline of stocking, and if you're spending some money to get your system up and going, it's really not that much compared to everything else that is going into the lake.
Yeah.
And this actually looks at the fish. This is going to tell you what you have and what you can do to make it better.
Well, on the private side, we're just barely learning how to use this technology because prior to Red Hills, what I could do, Bailey, is tell you if you had Florida, Northern, or some sort of intergrade, which is what we call a cross between the Florida and Northern. That's all I could tell you. And now, for $1,500, during an electrofishing survey, I'm going to collect more than 35 fish. And so we can even narrow this down further because we can say how related are these fish. So just pick 35 random fish out of your population. How related are they? Which may tell us, do we need to stock new bass genetics in and diversify this population? Josh will also tell me if those 35 fish have trophy markers. So I can look at the 2 to 4 pound fish and say how many of them have the probability to get above 8. Or I can even look at juvenile fishes of the year and see who's contributing most to the spawning class, like which fish are most efficient at spawning. So there's so many different ways for us to look at a private fishery now. And what Josh is telling me is we can do the same thing for public reservoirs and public water bodies. But first, we need to do the big test. We need the million marker test. So this is going to be a fun adventure that you've signed yourself up for, Bailey.
I'm all about this. This sounds exciting as hell to me. And I know there's a line of people spread across the north that would be jumping at the opportunity to do this.
Really, that's great to know. I've been in the southeast for a long, long time. My wife is from Long Island, but we don't get up there that often. And to know that there really is on the ground interest from an angler perspective, that everything else follows what the anglers want to do. So that's great.
Yeah, I think you ask anybody up here, that fishes avidly, and you ask them if they'd be interested. I think they would be very excited and say yes. Anything they can do to help with that, I think they'd be, ecstatic to do it. I think for a long time though, it's just been like they don't know that this is even a possibility, which is cool that we're showing this throughout the show, or even how to get involved in something like that. There really hasn't been a way in the north, because there hasn't been studies done.
Sure.
So I think if you present this opportunity, you find a waterfall of anglers that are willing to participate and help.
Bailey, tell Josh and I what's a trophy northern bass? Like, what size should we be targeting?
I feel like largemouth wise, anything. I mean, a 5 pounder is a great fish, but I think anything over 6 pounds largemouth wise in the north is a great largemouth. I would consider that definitely of a trophy caliber where you're not going to go out and catch one every single day, where you have a decent chance to catch one close to 5, if not over 5 about every day. I would say 6 pounds on the largemouth side and then 5 on the smallmouth side.
Okay. Yeah. So we'll push it. I mean, I don't know. We'll push it a little bit above that because the same thing with what you did for Florida is you want to figure out how do I grow a 15-pound Florida. You don't start with 5-pound fish. You go above what is common. So we'll find that number and put this together, Bailey. This is going to be a great experience. Now, Josh, what is the likelihood? I'm going to put your hypothetical cap on. What is the likelihood that some of these fish, even in New York, are carrying a Florida marker that we didn't know about?
It's a possibility. That's the only way that I can put it. I don't know specifically the stocking history of, the upper Northeast, those areas. I know that fish have been transferred all over the place for quite some time. I mean, the Japanese fish that we looked at, last year is 100% Florida, the world record. So Florida's made it all the way to Japan. There's a very good chance that they've been dribbled in other places, and there's no record of that. People could have caught fish in Florida and transferred them back up to New York or Massachusetts or wherever a long time ago, and nobody would know it.
Yeah, that could be a possibility. So one thing we might learn is that our northern fish that we were hoping were pure northerns and growing above, hitting that 8, 9, 10 pounds are actually partially Floridas. And once again, we call those integrades. And I think there's whenever it comes to anglers and their desires, we hear a lot about F1 bass. And I think before we get into Kurida's fish in Japan and all those things, it would be great for you to explain what an F1 bass is.
Sure. So F1 is just is a generic genetic designation for anything that's the first generation offspring. So if you have two parents, their offspring are F1s. If you take those offspring and do something with them, their offspring are F2s and F3 and F4 and F5 on down the line. For an F1 in our community, an F1 is typically a Florida female that's crossed with a northern male. And then so you have the offspring are 50-50. It's a combination of both. There's all sorts of different thoughts about what F1s can do within a population. Genetically, you're really rolling the dice. And this might get really into the weeds from high school biology and genetics. But if you look at what's called a Punnett square and just do the math on how things would move through to offspring, you've got the Florida fish, which gets bigger, is less aggressive and doesn't grow as fast, and you cross it with a northern, which grows fast, is more aggressive, but doesn't get as big. All the offspring that are of that cross are not going to have exactly what you want in there, which is fast growing, gets big, and is more aggressive. In fact, you're probably going to have the same amount of offspring in that F1 generation that do the exact opposite. That grow slow, don't get as big, and are less aggressive. And that's because of the math behind it. You're taking three opposing traits and crossing them. The majority of the fish are going to have a combination of that. So where you might have 10% that do get big and do everything you want, 10% that don't do anything you want, 80% are going to be a combination of those traits in the middle. So if you're stocking straight F1s, yes, you are likely increasing the opportunity by 10%. But over the course of time, because you've got all this mix mash of genetics, as those things start to breed together, the things that you want are probably going to start to wash out over time. That's not to say there's not a place for stocking hybrids, but it's, from a straight genetics and math standpoint, it's not as straightforward as just crossing them and all the offspring have what you want at the end of the day.
Yeah, so you go back to that Punnett square. You start talking after you go the F1s and you have this mixed match of genetics. You do the F2s, the F3s, and on, and you're actually reducing your likelihood of those desirable traits, right? You have to then reintroduce those desirable traits from another source, from a pure Florida or something like that. So, I mean, this is definitely something on the private sector side that we recognize. If you want a trophy fishery, a true trophy fishery, pure Florida offers you the best opportunity at that. Now, there is a caveat that there's a northern limit to what pure Florida, especially juvenile fish, can be stocked. Whenever we start talking about the stocking process, using fingerlings most often, and fingerlings have a high mortality over the winter for Florida as you reach a higher northern latitude. You get a slightly larger fish or even an adult Florida, and the success of them integrating into the population is much higher just because conditionally those fish can survive in the environment slightly better. So when we're talking about moving fish and stocking fish and F1s, there's a lot of caveats to should this be done. And, we can't, for the betterment of fisheries, we can't leave this conversation without saying we also don't know a lot about regional genetics. And just like Josh was saying about Florida, the introduction of certain fish would have changed that population and maybe something we could never go back to. So getting these studies done is extremely important. So there's big competing, desires for bigger fish or for better growing fish, but we also have to balance that with what is best for our fisheries long term and preserving natural genetics.
Yeah, and going back to the F1, and I mean, really any of them, once you stock fish, then Mother Nature starts to select who the winners are going to be. So if you take Floridas and stock them up north, and then wait 20 or 30 years, you're going to start to see less and less Florida genetics within the population to the point where they're probably going to get washed out after a while. For the hybrids, they're half Florida. So what you see not only is this mashup of genetics over time, but you also see if you stock those in, let's say, Illinois, where you've got really cold winters, over time you're going to start to see Mother Nature select against the Florida genes, the ones that you want to grow big fish. Those are going to start to get weeded out naturally. And what you have at the end of the day is probably going to be very close to, 90% northern genetics after stocking 50-50.
Amazing. Let's see, Josh. I want to talk about world record, but I don't want to leave without finishing and wrapping up this Red Hills bow. Y'all's offerings right now. If I go onto the website, I can purchase a genetic kit, which would tell me Florida versus Northern versus integrate, all the things we just talked about for those 35 fish, along with relatability, so daughters, granddaughters, cousins, all that kind of stuff, and trophy markers on the Florida side. And then what if I just want to purchase fish? What kind of bass do you guys have for purchase?
Sure. So we've got 4 different lines. We've got straight Florida, straight largemouth. We do sell hybrids because there is a call for those, and they do have their place in certain areas. And then we sell the Max line of fish, which is the pure Floridas that are bred from trophy marker parents. And typically, what we tell folks is, yes, you can reach out to us to purchase fish, and we do have some partners that are working with us to do this air freight. But really, it's the best thing to do is to consult with a lake and pond manager at the end of the day, because I get a lot of phone calls about what should I do for my lake, and I have to say I really can't advise on that. I've never even seen your lake. I might know what state they're in, but having a lake and pond manager who has the experience, can read water, knows what parameters to look at, consulting with them, even if it's not a long-term deal, but just having somebody come out to talk about your system with you before you stock fish, I think is probably one of the best things that you can do.
Yeah, I love that and appreciate that because that feeds our business. So we love that. What about not private lakes? What about state agencies? Is Red Hills seeing an uptick in state agencies wanting to participate and purchase fish or do genetic work with this?
Absolutely. So we do some work with various states as far as fish offering, but our most states have got hatchery systems, so they don't really need to purchase fish. What they need help with is genetics, and that's coming from a state agency. That's one of the biggest things that I think is important for us is working with the groups that are conserving populations, managing populations, to help them do better with the genetics of their fish, to learn more, to incorporate that into their hatchery systems. So we have a number of different states, and we're talking with more and more as we kind of get more of this research online to really help folks, from a regional standpoint, not only get better fish, but to be better at what they're doing from a hatchery system standpoint too, and incorporate that into there.
Man, Josh, you got a lot going on. I think
Yeah.
it would be a terrible miss if we didn't talk about your work with world record genetics and the Karita fish and just the whole story. So I'm going to give everybody a little background.
It's our work, Stephen, by the way.
Yeah. Okay, thank you for involving me. Let's see. A little background. George Perry, sometime in the early 1900s, claims to have caught a 22.4 pound bass. We've never seen a picture of the fish. There's no official record. That fish was submitted into a fishing contest on Field and Stream magazine and has been accepted as the world record for most of this audience as long as we've been alive. A second fish was caught in the early 2000s in Lake Biwa by Manabu Kurita. I think he was fishing with a live crawfish. So some people would say, well, he's fishing with a live bait. That doesn't count. But it was certified. It's actually probably the most certified fish that has ever existed whenever we talk about world records. But it was certified by IGFA at 22 pounds, 5 ounces. So 1 ounce bigger than Perry's fish. But because of their rules, you have to at least have 2 ounces to be the world record. So those two fish have been tied since the early 2000s as our world record for largemouth bass. Now, there was a 25-pound fish caught also in the early 2000s, but that fish was actually foul hooked. Whenever they were fishing with a jig, they were sight fishing, fish was caught in the side. So they did weigh and certify that fish, but it was released and has never been considered a world record, although it's the biggest fish on record to ever be caught whenever we talk about largemouth. 2 years ago, the IGFA followed American Fisheries Society and split largemouth and Florida bass into 2 species. And when they split them into 2 species, through some recommendations by a committee, they decided that both Manabu Kurita's fish and George Perry's fish would actually go to the largemouth bass record and vacate the Florida bass record. The Florida bass record was then certified as a Texas fish that weighed a little over 15 pounds, caught by Kyle Hall out of OHIV, because that was the first fish that could meet a standard of pure Florida. And then Josh got involved. And Josh, along with our friend Ken Duke, hunted down and found the skin mount of Manabu Kurita's fish. So the fish has been a skin mount for 20 years. Happened to be on exhibit in the Yamaha facility in Atlanta, Georgia, right outside of Atlanta. And Ken, Josh, and I don't know who else was involved in negotiating this deal that you could go in and collect a sample off this fish. But you led a team to go sample this fish, and I would love for you to just walk us through what happened next.
Sure. So we had access to the fish. We knew that was going to be a special one-time thing, but it was very important to do it properly. So one of the things we considered was we really need 2 independent labs to do this. And through connections with Florida, we got Brandon Barthel to come up with us so that Florida's lab could also run tissue samples completely separate from us. So we gained access to the fish. We were able to collect tissue sample from the fish. Florida went back to their lab. Our lab is in San Diego, so we sent tissue samples off to San Diego. And then we waited and let the labs do their work, which was really the biggest question. Can we get any sort of usable DNA off a skin mount that was heavily lacquered and is 20 years old? Both labs worked very hard at that. In this whole process, the first thing you do is extract DNA from the tissue, and then you test that to make sure that it's quality enough. Both labs probably went through 3 or 4 different rounds of trying to do that. Our lab was able to get probably about 60% of the DNA that they acquired to work, whereas Florida was able to get 100% of theirs to work. And both groups, it was their Hail Mary play. You know, both of them had said, if this doesn't work, then, we're done. We can't do anything else. But the cool thing was that, we got our results back, and I think about 60 to 70% of the markers actually amplified. Those said that the Carita fish was a straight Florida. Separate from us, like I said, Florida got 100% of theirs to amplify, and they said their fish is a straight Florida. So Florida, because they had really good results, they are the ones that kind of took the lead, submitted the test back to IGFA, and we got the record changed to where we knew it should be, which is a pure Florida fish for the world record. Unfortunately, Perry's fish, that's been long gone. There's nothing we can do there. But the official world record, the one we all know existed, is a pure Florida bass, and it is now categorized as such. The cool thing for me was that this was kind of a way to do more work on the Carita fish, right? So we've gone in and we've said it's a pure Florida bass, but right now we're currently running that fish's DNA in another very broad trophy project, along with about 500 other 8+ pound fish out of the state of Florida, so that we can refine and maybe see some additional things that we didn't see in that pilot study we did a couple of years ago.
So, wait, go back to this Jurassic Park thing. So you're telling me that what you're going to actually do is you're going to find markers that Carita fish have, and then Red Hills is going to sell Carita fish offspring.
Correct.
See, that's the kind of Jurassic Park I can get behind. As much as I love dinosaurs, let's create dino largemouth. Let's do that.
Bailey, this is really cool research, because for the first time in our known history, what we can do is we can take those old skin mount fish that we've all seen. They're falling apart. They're not doing great now. And we can take a step back, hit a time machine, and go back 30 years and say, what were the genetics? So you go to the pawn shop, in Coleman, Alabama, and you find an old skin mount, and you know the history of that fish. All of a sudden, you could say, let's look at what grew this fish, what genetics was this fish, and we could start doing that. So just like your northern, where we're saying, let's go look at what's happening in Northeast right now. Well, if we can find those skin mounts in somebody's basement and know the story of it, between Florida and Josh, we have a system now to look at the genetics of those fish. So not only did Josh change the world record to its rightful place, we knew that Carita's fish was going to be a pure Florida. Pure Floridas were stocked in Lake Biwa in the 80s and maintained. So we knew that was going to happen. Myself and probably Josh both agree that if Perry's fish existed, it would have been a pure Florida as well. That's just what's most likely. But Josh proved it. Josh proved the Carita side. So congratulations to you. You are part of a team that changed the world record. Very few people can say that.
That's awesome.
And I think, what did I say? I think I said this on Ken's podcast, or maybe I said it to Ken in a sidebar. That is the only fish that I believe has held 3 different world records as a singular fish. It held the largemouth as one species. It held the largemouth as one of 2 species, and now it holds the Florida.
Amazing.
Crazy.
So Josh, what's next? What's next for Red Hills? Are you going to change this world record again by growing one yourself? Like, where are we headed?
Well, so we're doing a lot of things, like I said, behind the scenes, continuing to do some pretty significant research, and a lot of that we're working on now is probably bigger than what we've produced so far. Some of that has to do with the Karita fish and taking a deep dive into that to see what its genetics are. We're also looking at, the other traits of interest, which is aggression. We're still working on trying to get a genetics marker for gender, but we're also looking and diving deep into the ability to produce all female fish, which we think is going to be a huge game changer if we can significantly produce all female fish, and not just a couple thousand a year, but tens or 20s, 30, 40,000 fish. Not only does that increase the opportunity for people to catch big fish, but it also brings on a new management aspect, because if you can limit the reproduction or eliminate reproduction in a system, that's half the battle in fisheries management, is making sure that your population's fed and it's not too crowded. I think there's some exciting things there that are going to be coming out probably over the next year. But going back to what you said about these old skin mounts, what's really interesting to me is looking at historically having the ability to see what genetics were 30 or 40 years ago and see how they've changed, and see if we can put them back, if we should put them back somehow. And not only to protect species or populations that were there, but also, I don't know how many times I've talked to people that have been around for quite some time and said fishing was better 40 years ago. Well, is there a reason? And could that be genetics at the end of the day? That to me is kind of the big thing, is looking at that, taking a historical approach to genetics.
That's cool.
All right, Bailey, you got anything for Josh?
No, I'm just kind of. I keep replaying the Jurassic Park thing in my head and just wondering what does bass fishing look like, in 10 years and 15 years and 20 years, going down, this trend with you guys at the helm. Like, it's exciting, man. Like, just to see the things that we can. Oh, I should not use the term we, but you guys are doing, and we as anglers can be excited about. So it's pretty dang cool. Literally creating a bass Jurassic Park,
but I think there is a
child in me is nerding out about.
Bailey, there's a we component. We heard it today.
Absolutely,
there's a whole side of this that we need the anglers for, and you could be the tip of the spear. You could start kind of the process of how do we look at northern fish the same way.
You say when, man. I'm in. Awesome stuff. No, seriously, this has been really cool, Josh. I'm excited to dig into this further with you. And
sure,
this is just episode 1 here with you on this show, man.
Cool. Yep. Well, I look forward to doing more, and we'll talk more about how we can get you guys some test kits to start getting some data.
Heck yeah.
All right, awesome. Josh.
I'm sure when this episode goes up, I got a bunch of buddies in the north, not just New York as well, that listen to this, and they're going to be like, sign me up. Just like opposite of this pretext. I'm sure I'm going to get bombarded with that.
Great.
Heck yeah. Steve, what else you got, man?
That's it. I really appreciate you, Josh. Of course, I love you as a vendor. You guys produce great fish and always help me out. We've got amazing projects that we've been working on. I don't know, it seems like 4 or 5 years now. Stocking your fish and seeing the results, and we're always so happy to work with you. Love the genetic test. It's really opening up opportunities for us. And I really enjoyed the Karita project too. That was a fun way for us to interact in a different setting. And I'll never forget us walking out of the building with those samples. And we were 12 years old at that point.
We all 3 of us got in the truck and giggled.
Yeah. 3 grown men giggling like we just pulled off the heist of the century, but it wasn't. Exactly. It was I think a big step in the scientific process of understanding our history, and we'll see how that can play out in the future.
Absolutely. Well, there's a lot of work left to be done, and both of you guys, I look forward to working with you on stuff going forward.
All right. And the next episode that we do together, I want you to be prepared to answer a very difficult question, which is what percentage genetic material does a fish need to be a pure something?
Maybe give me enough time. Maybe we'll have an answer for that.
That's exactly right. All right, brother. Thank you so
All right.
much.
Thanks, guys. See ya.
See you, Josh. All right, folks. There you have it. Basically, look forward to 2030 Jurassic Park Largemouth Bass Edition coming in hot. Maybe a smallmouth bass edition potentially, but some pretty awesome stuff unleashed by Josh, man.
Yeah, and we're gonna have to have Josh back on because we didn't even get into smallmouth bass genetics. I know he focuses on Floridas and largemouth, and I think a guy that knows as much as Josh does, there's some opportunity there for us to chat smallmouth and really learn if there's any way to keep growing smallmouth to that next level too.
That would be it's already booming, as it is with just nature being nature. I think that's the Gobi effect, the what you call it, the not mussels, what is it? Oh my gosh, whatever the thing that clears up the water, you know what I'm talking about?
Zebra mussel.
Thank you. I'm just not brain's apparently not in it on this Friday morning here.
Oof. Well, you did hear a lot of science today from Josh, so I'm sure you're on full download right now. You get the excuse.
I'm still picturing just opening up the stereotypical giant Jurassic Park gates with a Jeep going in, and it's just bass instead.
Yeah,
brain still fried from that, but yeah, no, it'd be pretty wicked to see if just with some testing and some things, research being done behind it with little to none being done besides our own little angler theory up north of what could be done with even smallmouth, and
right.
Start building out 10-pounders in the north, because there was one caught in Lake Erie. Was it last year or 2 years ago? There was a 10-pounder caught in Lake Erie. I think that's now the new Ontario record, I believe, was claimed. I'm trying to remember how the grid worked, but I think it was either Ontario or Ohio, one of the two. New York kind of got the shaft on that, but it's okay.
It's okay. But you guys have had some great fish the last couple of years. You can give one to another state. Or
I feel like we've had a state record every year for the past 4 years. So it's just going to keep going, and it'd be cool to see, guys like yourself and Josh with the brains to see what could happen, not just in New York, but like Michigan. You look at the UP and they got some mega smallmouth up there, and across the states from Minnesota, Wisconsin, all that largemouth smallmouth would be cool to see what we could do with the North. But man, it's just a great episode, and I think without further ado, let's get into the Lake Hotline question we have for this episode.
Oh yeah, Lake Hotline. Let's do that.
All right. So for folks that maybe this is your first episode, Lake Hotline is where you guys submit your questions direct for Steve and or the guests to answer on a show. So if you have any questions you'd like submitted to Steve, like we said earlier, send us a DM on social, send us an email, comment in the YouTube comments, and chances are we'll submit your question here, Lake Hotline. But for this show, we had a comment on the episode that we did with Troy. This is a comment from MJS Bull. Said, great episode, gentlemen. We'll break this up because I think there's a couple questions in here.
Okay.
Said, I do have questions. Would like to hear your opinions and thoughts on treating small bodies of water. They use an example of less than 10 acres compared to larger bodies of water, 20 to 50 acres of water, and how you would, I guess, differ from treating those different bodies of water based on size.
I got it. Yeah, so Troy Goldsby was talking about aquatic plant control during his episode, and so I'm going to assume that's exactly what we're talking about here, is doing some sort of treatment protocol on a small body of water versus a large. So number one, the smaller the body of water is, the less forgiving it is for mistakes. So whenever we start talking about plant biomass, we really need to quantify that well, and then we need to figure out how can we control it without getting the detrimental effects of the degradation of that plant. As it starts to decompose, it's going to use more oxygen. You're going to have slight water quality changes, especially whenever we start talking about the off gases. So if oxygen's present, carbon dioxide. If oxygen's not present, then it starts to switch to ammonia. So you could really, in a small body of water, you could get yourself in a bad situation because you spray too much plant biomass at one time. So whenever we're talking about small bodies of water, we want to really limit the surface area or the biomass of plant that we're trying to control at one time. We want to make that a smaller amount so that the pond or the lake can digest it. And you will see a lot of times water color changes in those small bodies of water, because once again, that environment, it just can't handle rapid change in water quality. A larger body of water is usually more forgiving. At the same time, a larger body of water, we would hope, has a lot of the area of the body of water that is deeper than the littoral zone and is naturally controlling vegetation just due to depth. And so the larger bodies of water, you should be able to use a protocol that's slightly more aggressive, as long as you have some of that open water refuge. So hopefully that helps with the small versus the large body of water.
Awesome. And the part 2 of that is they mentioned they really liked our conversation on utilizing artificial cover. And say for lakes that have a decent amount of vegetation, things of that nature, if they're struggling to produce vegetation and there's not much cover in that lake, utilizing artificial types of cover, what is the most beneficial on those smaller bodies of water?
Yeah, also a great question. So whenever we have a lack of vegetation, provides some specific niches for fish, especially juvenile fish. It provides an escape habitat. So we look, vegetation, as we look at it, it creates these dense mats that allows juvenile fishes to go in and out and escape predation. It also gives them some area to forage around where insects are growing on that vegetation. And then for our larger fish, they use that vegetation, the outsides of it, to ambush forage. So what we need is some sort of habitat that mimics that. That would be high dense habitat. You could think of if you just cut like a cedar tree and set it in the water. That's very high dense and would serve some of those same purposes. The problem with just cutting a cedar tree and setting it in the water is a cedar tree is going to last. Needles will be gone within 18 months. Stems will start to degrade and be gone within 3 years. So it's really short term. So instead, we often select artificial habitat because artificial habitat, once it grows algae, it mimics natural habitat, but it lasts for 600 plus years. So we really wouldn't have to replace it in the future. There are several brands of commercial habitat that give you that high density. One of them that I helped design is from a company called Mossback Fish Habitat, and we call it the Conservation Cube. The Conservation Cube is these PVC tubes, 4 of them, and then they have interlocking V-shaped limbs that connect those tubes and make them into a cube. And then there are about a dozen internal limbs off of each one of those tubes to create a really high dense center that would mimic vegetation, would mimic that brush pile. But what you're looking for is that high density. It allows juvenile fish to go in and out. It allows them to graze for forage, insects mainly, and then allows predatory fish to use it as ambush habitat. Now, density of the habitat is one part of it. The number of those habitats is the other part. Aquatic vegetation covers a large area quickly, and you really want, somewhere between 10 and 20% of a water body to have habitat. So if we don't have any vegetation growing, we're going to need a lot of artificial habitat, and that's becomes one of those situations where you may use some of the cedar, some of the artificial, maybe even some rocks or boulders, and you use multiple types of things together so that you can get, in an affordable way, enough habitat in the pond.
Awesome. Well, there's a lot of info there for you. That, I think, answered the question tenfold. But it's cool to see how the smaller bodies versus larger bodies of water differ, and how, at least your approach, Steve, into creating a better habitat for the fish, but also a better place to fish for the angler.
Yeah, and that's the hard part. Whenever you build something just for an angler, you leave out why are fish attracted to it. And whenever we start talking about like our major league fishing projects, I really always try to focus on, let's attract forage if you attract forage to habitat, naturally you get predators. And when you attract predators to habitat, naturally anglers catch more fish around it. So it kind of works in reverse rather than just putting out a single piece of structure that will hold fish temporarily, but maybe doesn't change the environment because we don't have enough density or we don't have enough of them.
Awesome.
Yep.
Well, there you have it, folks. There's your lake hotline for today's episode. Make sure you guys submit your questions in for Steve. And man, is there anything else we need to cover in today's episode?
I think we're doing great. Real Biology has really hit its stride. We've had some wonderful guests. We've got really great guests lined up as well in the coming weeks. Can't wait to have some of these discussions as we move to, growing bigger bass. How do we manage them? Learning these fisheries techniques. Hopefully, we're doing it in a way that everybody can understand. We see some common themes across these episodes where we're almost always talking about harvest. We're always almost talking about habitat. We're now almost always talking about genetics. You're seeing that no matter where you are in the country, hopefully you have seen or will be hearing from a biologist who really cares about your fisheries, and it's always common themes. So these episodes are going to be cumulative over time. You're going to continue to learn fisheries management. I'm just so happy that our audience is along for this ride.
Heck yeah. I think with that too, it'd be a really cool call out, Steve, to basically just mention to folks like, submit your comment or question or whatever, hailing from the state you're in. I would love to hear where one of you're listening from to, talking about, your state department. And if, hey, like, reach out and request to have somebody from your state on these shows. We'd be happy to oblige for that. Not saying we're going to make it the next episode, but it definitely will something that we can factor into the lineup. If we have people reaching out like from Minnesota, and they want to talk about something specific from Minnesota, like that's something we will heavily take into account, and we'll love to hear your guys' feedback and where you guys are listening from. So I think with that, Steve, and we wrap this sucker up, put a bow on it, and appreciate everybody for tuning in. Appreciate you always lining up these shows for us and bringing out the juice and our guests, man.
Well, I'm just lucky to have a good network, and like I've said in the past, there are not biologists that aren't anglers, and so it makes my job pretty easy. Let me find the great ones that are doing fun projects, and let's talk fishing.
Heck yeah, man. Well, awesome. Appreciate the folks for tuning in, and we'll be seeing y'all on the next one.